The final segment before lunch is to give an update about this MRD question and as it relates to the Black Swan Research Initiative, which is really focused on this. And so I hope that this will be of interest to all of you. I have a slide presentation, and then we have a very short video just to show you some perspectives on the Black Swan project. And so you'll see that we have brought the black swans to Florida, especially for you. We have the black swan, and you can see that even some babies came too. Those are the new research projects that are coming along. What is the purpose of the Black Swan Research Initiative? Firstly, it's a global initiative, and the idea is to get contributions, ideas, research projects from all around the world to focus on this and have this as a global project. And it is focused on using a precise method to measure MRD. If we're going to say that, as Jean-Luc just did, is that for the future, moving forward, looking at the level of myeloma at this low minimal residual disease level, if that is what we're going to focus on, then it better be a good method. And so that was the first target for this project, is to try to have good, reliable methodology. And so it would allow us then to be comfortable that if we get an MRD negative result or a positive result, this would be sufficiently reliable that we could take action to either continue treatment, stop treatment, or maybe even change treatment. So you could see, if you're going to make those life-changing decisions, it better be a darn good test where you're comfortable at those very low levels. And these treatment decisions are things that need to be evaluated within a clinical trial. And right now, to get to this lady's point over here, outside of clinical trials, we don't really know how to interpret either a negative result or what to do about a positive result. And so the major goal is that if you have myeloma which is at this 0 MRD level, where there's less than 1 in a million cells there, then could this translate into cure? And that would be an extension of this, where not only would you achieve an MRD negative, but it would be sustained. And within this project, as you'll see, it would be sustained at 1 year, 3 years, 5 years. And so it's a sequential thing. It's not something right now where we could say, yes, you got to MRD negative, you're cured. No, this is something right now where we would have to follow that and see if it is actually sustained. At the level of 1 in a million, it looks like it would have to be sustained. It's maybe if we got down to 1 in 10 million or 1 in 100 million, if we got it down maybe a little bit lower, it could be that just one measurement would be enough, and we'd say, oh, well, you're down at that level, that has done the trick. So the first goal was to have a reliable test. Now, when we started, there was one testing available, which was the sequencing method being developed by a commercial company, Sequenta, and they were looking at a method, looking at the DNA in the myeloma. We wanted to try to have a test that would be more broadly available and less expensive, but equally sensitive. And so we set up a research project with the team in Spain, in Salamanca, Spain, with Professor Jesus San Miguel and his team there, Bruno Paiva and Alberto Orfeo, very much focused on this immunophenotyping that Jean-Luc mentioned, where they were looking at the immune typing on the myeloma as a basis for identifying the myeloma. What were the things on the surface of the myeloma that could be used to detect a myeloma cell versus a non-myeloma cell? And so they developed a cocktail of 8 different antibodies, which I'll show a little bit later when I'm talking about this. So they used 8 antibodies which identified the myeloma, and at a sensitivity of 1 in a million. And we're looking at cross-validation using even 10 antibodies or 10 colors. And this is also linked in with a computer system where the results go through the computer, so you don't need to be having someone look at these multi-dimensional colors and try to figure out what's going on. And so this software approach is something new to this technology. Up to now, when samples go to the lab across the US or around the world, it's still analyzed by someone sitting in front of the machine to analyze the outcome. And so making it into an automated software system is a huge step forward, and this is now possible with a rapid software that can run in 12 minutes. The question is, how did this system compare? And these are some of the early results. As you would realize, the patients who have MRD 0, that's the minority of patients, so that to get a sample, which they now have of 100 patients who are MRD negative, where you can compare this flow method, where the cells are run through a flow cytometer, versus the sequencing method, it takes some time. But this just shows you, in the initial testing, the problem with the NGS is it's not possible to run the test for every patient, because the DNA doesn't work well for every patient, and it was only possible to do it in 78% of the samples. Conversely, when they looked at whether one test was positive or negative, 21% of the time the flow method was actually positive when cells were not being picked up by the sequencing methods. And so this gave them tremendous confidence that the flow method was actually even more sensitive than the sequencing method. Now, in addition to this, if this is really going to work, right from the outset, the team realized that to make this available within trials, this needed to be a global effort. There are trials going on in the US, but there are lots of trials going on in Europe. There are trials going on in Latin America and in Asia. And so if this is going to be something where we can compare all these different trials that are going on around the world, it needed to be a methodology that could be achieved globally. And so the Black Swan team has been setting up and evaluating labs to do both the flow and the sequencing methodology. You can see in the US, there's at least half a dozen labs who are set up to do it now, and in Brazil, and in different parts of Europe, as well as Asia and Australia. To take these results and convert them into a standard response criteria, up to now, we're using the standard response criteria, which is very good partial response, complete remission, stringent complete remission. MRD is going to be the one which is beyond that. And so the idea is to integrate that into a completely new system. And so Dr. Shaji Kumar has been working with all of the members of the working group to come up with a standard way to do this. And what I've simplified it here just to be concise, but basically the new criteria, or MRD negative, is if you're negative by either this new flow method or the sequencing method, and if it's sustained. And then there's an additional category where, as you know, the myeloma might be in the bone marrow, but it could also be outside of the bone marrow, and that would be detected using a scan of some sort. And so the third category, C, is if the PET CT scan would also be negative. And so if you had A or B plus C and it's sustained, this would be something that could lead to a cure. Now, in addition, there are several projects as part of the Black Swan Research Initiative. I'm going to just give you a few examples. There are over 20 projects going on right now, but this one is where, to understand if you're achieving MRD, we're biopsying patients where there's a lesion which has not gone away. And so the team that is working on this is with Dr. Jens Hillengass in Germany. And so what he's doing is he's using the imaging to guide the biopsy. So if there's a big lesion in the pelvis, he will do a biopsy at the beginning to see what it looks like in terms of the genetics and the immune testing and the drug sensitivity. And then he'll do a biopsy afterwards and see, well, what's the pattern then in terms of the resistance or the genetics. And so this is a very, very key project to understand why are cells resisting initial therapy? What is the special aspect of the resistant subclones? So this is what's been accomplished in the last 2 to 3 years that the Black Swan project has been going on. Basically, it was thought about in 2012, started in 2013. So it's a relatively recent project, but even within this time span, all these projects have been up and running. What are we looking at now? What are the next steps? So, as was touched on in our discussion here, we need to understand all these different aspects so that if you have an MRD test and it's negative, we need to understand what does that mean. Is it safe to stop the treatment? Should you continue the treatment? And so we need studies to look at that, where half of the patients would continue the treatment, half of the patients would stop the treatment. When we look at other diseases, strangely enough, the patients who might benefit the most are those who are MRD negative and perhaps continue treatment for another 3 to 6 months to get rid of these last few cells. And conversely, if you're MRD positive, the treatment's not working, and it would be good to stop the treatment and use something else. And so these are the things, the little details that we need to understand within clinical trials before we can make recommendations overall. The MRD positive part, if there's residual disease, this is where a whole range of projects are going on right now to understand the nature of those cells and come up with different protocols to evaluate new treatments. As was mentioned, it could be possible to do this in the blood. As you know, doing the bone marrow, if we say, okay, we're going to check the MRD, and then, oh, maybe we didn't get it quite right, we're going to repeat the bone marrow. That's not so cool. And so it's going to be tremendous if we can have a blood test. And so the first thing that the Spanish team has done, and also the team at Mayo in Rochester with Dr. Kumar, is to look at using this next generation flow method to pick up myeloma cells in the blood. And it really has led to a little bit of a different understanding about myeloma, because you can see here 100% of patients with both myeloma and smoldering myeloma, they actually do have small numbers of plasma cells in the blood, which we hadn't really been thinking about. But this is really why myeloma can become multiple myeloma, is that there are small numbers of these cells floating around. And now, using this test, we can see how many are there. And this is the next aspect of this study, is to look at how many cells are there, and this could be a way to evaluate the disease. But another group, and this is from studies being done by Dr. Andrew Spencer in Australia, he's looking at DNA that floats free in the blood. And this is amazing also. The DNA is released from myeloma cells that are getting destroyed spontaneously or by treatment. And you can pick up that DNA and you can study that DNA. And you can look at what are the mutations that are in this DNA. And at ASH in December in Orlando, Dr. Spencer, he showed very different kinds of mutations that are showing up in relapse. We think about high risk with this 17p. That actually does not seem to be so important at this point of later relapse, where these different mutations, which are called RAS, you can see the different colors here. NRAS is a blue one, and KRAS is an orange one. You can see that these different mutations in the RAS system seem to be more dominant. He studied a panel of 96 different possible mutations, and these are the ones that came to the top. These different researchers, right now we have over 40 researchers in 12 different countries looking at different aspects of this problem. And this has just been amazing to see all of the results. It's to the point now where we can have a meeting of our own. Just on Black Swan, you can see all these different researchers have got lots of things to show and to talk about. The first thing that we're really excited about is that we're starting these trials to really make this first effort to cure myeloma. And the CAESAR trial, which is in Europe, is coming from Spain with Dr. Maria V. Mateos and Jesus San Miguel starting this trial. And the Ascent trial, which will hopefully start in the US in the next 2 or 3 months, with Shaji and myself leading that effort. So what are those trials attempting to cure myeloma? Well, the CAESAR trial is an aggressive approach for smoldering myeloma, high-risk smoldering myeloma, where these patients are receiving, and about 10 patients have been treated in this study so far. They get carfilzomib, which is Kyprolis, Revlimid, and Dex for 6 cycles. They receive a transplant and some more Kyprolis, and then they have maintenance for 2 years. And so in this case, the idea is to have the treatment be about 2.5 years in total, and that would be a curative strategy with monitoring at 1, 3, and 5 years. And in the US, the Ascent trial, you can see the acronym, and it took a little time to come up with these acronyms. But anyway, in this case, you can see the big difference is that the daratumumab is added in addition, and there's also an option for transplant or not in the US trial, and we use the dara and the Kyprolis as a maintenance in our study. But again, a constrained amount of treatment. But what we're really excited about is to try to have an effort to achieve cure for patients who are not in this early phase. So for the broader patient with myeloma, the idea is to have a study for patients who have achieved a CR but have become MRD positive, to study those cells and come up with some treatment options for them. And so Dr. Rafat Abanour, we're just brainstorming with the ideas of the next trials for this population, but one idea is to look at different combinations of the new drugs for these patients with MRD positive disease, to study them and also to have different options of treatment to see which one can do the best job to get rid of the myeloma and convert it back to a sustained MRD negative. And we have already been looking at the pattern of disease. We know that the gene expression profiling is different. The cells which are left are not the same as the ones that you started with. The color pattern you can see is obviously different, and it's known that this could be related to resistance to Velcade, for example. The pattern of genes can show that they're resistant to Velcade. But in addition, we know that the immune system of the myeloma is changing over time. If you look at the plasma cells and the T cells and the other immune cells in the marrow, it's been noted that the immune pattern in the myeloma changes in patients who have successful therapy. And there would be an opportunity to enhance this by these immune therapies, especially those ones that are very attractive right now. And so this just shows you one of the current strategies of thinking of immune therapies that could be a key approaches now and for the next few years, targeting all of these antigens which are on the surface of the myeloma, using, for example, daratumumab, which is an anti-CD38, or elotuzumab, which is this anti-SLAMF7, or to use these cells which are called CAR T cells. So the T cells up at the top there, which are attacking the myeloma, but in active myeloma, their activity is blocked, so they don't work. So you can use a checkpoint inhibitor, or you could enhance these CAR T cells to make them active against the myeloma. And so there are quite a number of immune therapies which could be really crucial to get rid of the disease that's left and to attack it at this low MRD level, which is a whole new concept in itself. And so this gives you sort of an overview of where we are with the Black Swan Research Initiative. You can see that we have high expectations, that we are working in the blood now as opposed to the bone marrow. We're going to be looking with MRD positive trials in addition to our cure trials, and really slowly documenting the fraction of patients that are staying in this MRD negative status and hopefully can be cured. So it's great to be able to see this happening and to be able to use this word cure in a sentence where it seems like it might be possible. So thank you. And now we're going to show you a short video. So if me in real life is not enough, you can watch it in the video now. No, it's going to be mostly other people in the video.
The Black Swan Research Initiative is a project where we are trying to go for a very big advance in myeloma. We have seen many incremental advances, but the whole idea behind Black Swan was that we need to go for a dramatic improvement.
For the first time, we are able to treat myeloma in a more effective way. With novel combinations, we can achieve deep response. And we suddenly realized that the deep responses were so deep that maybe some patients could in fact achieve cure.
So establishing a tool to measure success of therapy, minimal residual disease negative state, was the first step.
To track the myeloma at very low levels, we needed new testing. We needed to be able to have sensitive and standardized testing to monitor the disease.
So far, in the last 2-3 years, we've really come down to a technique that can measure myeloma cells at 1 to 10 to the minus 6 level. Extremely high sensitivity. This is the next generation flow method. Now that we have a way of assessing how effective our therapy is, the next phase of the Black Swan Research Initiative is to try and see if we can develop a chemotherapy regimen that can potentially cure multiple myeloma.
We're so excited to be moving forward into phase 2. And the first step of the phase 2 is the start of what we call the CURE trials.
This is going to target patients with high-risk smoldering multiple myeloma and deliver the best available myeloma therapy to such patients and assess minimal residual disease or MRD negative state over time. The unique thing about the Black Swan Research Initiative trial that is planned is that it's not only using the best available tools to treat myeloma, but it's applying it at a stage where patients have generally not been treated at all.
For patients who do not achieve an MRD negative status, patients who do have residual disease after treatment, is that MRD resistant to therapy that we have been using? And so now we have multiple projects. We are so pleased to have several investigators from around the world starting to focus on the study of MRD-resistant disease. Why are these myeloma cells resistant to therapies thus far? What is the basis for that? And what new therapies can we use to overcome this resistance? What new trials do we need to evaluate this resistance and move forward to eliminate those resistant cells?
Up until this point, all we ever talked about was how would we make multiple myeloma a chronic disease that would be controlled. The Black Swan Research Initiative is our best effort to try to find a cure based on the information that exists today about possible paths to find that cure.
I just think that it deserves a lot of attention from a lot of people. I hope there are a lot more donors like myself, private donors that can jump on the bandwagon and help out. I know that the large grants are government and pharma, but I think as individuals it's a good idea to get involved and help because we're so close.
Donors are understanding that this is going to make a difference. This may be the project which makes a difference and starts to cure myeloma.
Just the thought that it's related to the word cure is so exciting and something that I really didn't think I would see in my lifetime.
Forget about what you might read in the literature from a few years ago. It's ancient history. We are going to get a cure. We are very close. It's just a matter of defining it. We might even be partly there now. We just don't know it.
That is really important for myeloma patients to be able to convert the fear they have when they were diagnosed originally to a hope that says, if not me, somebody I know or my children or my grandchildren will be able to be cured of myeloma.
What is going to be truly exciting is to start to document that we are achieving cures, that patients are staying in remission for 1 year, 3 years, 5 years, 10 years.
A cure was something that we talked about, but it was almost like a fantasy, a faraway dream, you know, like looking at some castle in the distance that we hope to get to. And now we know that the cure is coming, and that Black Swan Research Initiative will be the entity that will get us there.